Of this conference and give me an opportunity to speak to you guys and to share a little bit about my research that I've been doing in the Sutcliffe Lab. For those that aren't familiar with what we do in the Sutcliffe Lab, we're primarily interested in developing molecular imaging agents that target particularly cancer, and we use these probes then to image them with positron emission tomography. So today I'm going to be talking about my research with engineering antibody fragments to target the alpha V beta 6 integrin. So to give you a little bit of background, integrins are a large family of cell surface receptors that contribute to a multitude of cellular functions, including motility, survival, proliferation, as well as communication between cells in the extracellular matrix, as well as communication on the cell-to-cell basis. So if you look in the bottom left here, this is a cartoon of a typical integrin with its alpha and beta subunits combined in its activated form to receive both outside-in signaling, which is signaling from the extracellular matrix to the interior of the cell, as well as to transmit signals from the interior to the exterior in the extracellular matrix. Our lab is particularly interested in one integrin, which is the alpha V beta 6 integrin. And we're interested in this because it's overexpressed in several cancers, including ovarian, pancreatic, colon, and was most recently found to be overexpressed in breast ductal carcinoma in situ, or DCIS. The increased expression of this alpha V beta 6 integrin has also been correlated with a poor patient prognosis, as well as in a more advanced disease state. If you take a look at these middle slides here at the bottom, these are pathology specimens from patients with DCIS and stained for alpha V beta 6 integrin. And what you notice on the left two slides there is for very low-grade DCIS, you hardly get any very minimal staining for the alpha V beta 6 integrin. However, when you look at high-grade DCIS to the right there, you notice a much more pronounced expression of the alpha V beta 6 integrin, as noticed with a much darker brown staining present. Also, how the alpha V beta 6 integrin promotes this malignant phenotype is currently poorly understood, and right now there's no molecular imaging agents that have been approved for specifically targeting the alpha V beta 6 integrin. So our primary goal is to try to develop a very specific targeted molecular imaging agent to help improve the diagnosis of these cancers, as well as to help, hopefully, direct a more guided treatment approach to these diseases. And on the far right, you can see a Kaplan-Meier curve of also with patients with DCIS, indicating a much lower decreased survival rate with patients expressing alpha V beta 6 integrin, as indicated by this red curve here. So in order to develop this targeted molecular imaging agent, I've chosen to use the intact antibody as a framework for this. And the reasons are because the intact antibody has very high binding affinity as well as specificity for its target antigen. The intact antibody has also had much proven clinical success for therapeutics, and in fact, over 30% of the proteins that are now developed for clinical trials are classified as intact antibodies. So they definitely have proven themselves in the clinic for therapy, and I'm sure many of you have heard of the monoclonal antibody Herceptin that Genentech produces for treating breast cancer. However, to make the intact antibody into a molecular imaging agent, there are some things that need to be really altered about it, and some of these benefits need to be addressed. For instance, the large size of the intact antibody really makes sure that it hangs around in the blood circulation time for a long time. So this contributes to a very long biological half-life, and oftentimes unbound antibodies persist in the circulation for several days, up to even several weeks. So from an imaging perspective, this prevents us from really rapidly generating high-contrast tumor images between cancerous tissue and normal tissue. Fortunately for us, there's protein engineering techniques that we can use that really dissect the intact antibody and sort of whittle it down into its smallest fragments and then reassemble it based on the particular application that we're trying to use, as well as the pharmacokinetic properties that we desire. So if you look at this graph down here at the bottom, the smallest engineering fragment that we can produce is the variable domain, and as you start to move to the right, you migrate to a much larger and larger size with different binding affinities as well as different pharmacokinetic properties. So as you work your way from left to right, you have a small size and very rapid clearance, and as you work your way to the right side all the way to the intact antibody, you get very large size and very slow clearance. What's also very important though is when you're engineering antibody fragments, you really want to make sure that you're pairing the biological half-life of the antibody fragment with the radioactive half-life of your isotope that you plan on using. So for smaller fragments such as a single chain FB or the diabody, they're typically are very small and they have very fast circulation time. So you want to pair these with a short-lived radioisotope such as fluorine-18 or copper-64. However, with the mini-bodies, tri-bodies, and such, especially for the larger intact antibodies, you're probably going to be switching to a more prolonged radioactive half-life, such as copper-64 or zirconium-89. So for us, how can we take advantage of the pros of the intact antibody, the high binding affinity, the specificity, while still being able to manipulate the intrinsic properties of the antibody so that we get rapid tumor targeting and also rapid blood clearance, with producing the overall goal of being able to image within hours instead of days or weeks. So to do that, I've chosen to use the diabody format, and the reason being is the diabody is the smallest fragment that still retains the bivalent binding of the intact antibody. It also exhibits very rapid tumor uptake and very high specificity for the target antigen. So in this case, we can generate high contrast tumor images in the order of several hours. So how can we produce these diabodies and cis diabodies? Well, I've taken the intact antibody clone 6.3G9, which is specific for the alpha v beta 6 integrin, and removed the variable domains from the binding regions. I've assembled the variable heavy and variable light domain using a 3-amino acid linker. And what this does is it actually prevents the natural association of those two domains. And it actually promotes dimerization between a separate chain of variable domains. On the C terminus on the far right here, you can see I've appended a 6-his tag, which allows us to purify the fragment by nickel column chromatography. But to assemble the cis diabody, I've actually engineered a cysteine residue at the C terminus. And what this allows is, since the domains aren't able to come together naturally, this cysteine bond then forms a covalent disulfide bond between a separate pair of dimers. So the cis diabody you can envision as a covalent dimer, and the regular diabody is a non-covalent dimer formed by just a linker. We then take these two gene cassettes, and we subclone them separately into different expression cassettes to generate our stable cell lines. We've also appended a leader sequence that allows the protein to be secreted out of the cell, so there's no cell lysis to recover. We simply just recover the supernatants and then purify them over a nickel column. Once we've purified the diabodies independently on two separate columns, we want to buffer exchange the elution buffer into a suitable buffer for storage as well as for radio labeling. Once we've done this, then we concentrate them down using a centrifugal filter for long-term storage. So now that we've purified the diabodies and cis diabody, we also want to verify its size, purity, and identity. And we use two different assays for this. One is SDS-Page, and the other is Western blot. So on the left here, you can see the diabody in its reduced form, which you would expect as a non-covalent dimer. Elutes has a single band at 25 kilodaltons here. And with the cis diabody, since we have that disulfide bond, we can run it in reduced as well as non-reduced forms. So you can see with the cis diabody in its non-reduced form, we get primary formation of a covalent dimer at the 50 kilodalton band. We then want to transfer these diabodies from the gel onto a membrane and probe them with an anti-6-his antibody. And that gives us the verification that what we're producing is the diabody that does in fact contain the 6-his tags. And you can see we've done the exact same procedure here on the right with the Western blot membrane, using the diabody in its reduced form, again at 25 kilodaltons, and the cis diabody in its reduced and non-reduced forms. To further verify the purity and size of the preparations that we've created, we run size exclusion chromatography. And size exclusion chromatography is basically an HPLC column that separates material based on size. And we can look at the cis diabody here at the bottom, and we know we form a covalent dimer using that disulfide bridge. So we've got a single peak that elutes at around 23.2 minutes. We can compare this peak to their normal diabody, which is again a non-covalent dimer, and see that they elute at roughly the exact same time at 23.3 minutes. So this gives us a good indication not only of the purity, but also of the size of both of the preparations here. So now what we want to do is determine how well do these diabodies bind to the target that we're interested in, alpha v beta 6. So in order to do this, we run a competitive binding ELISA, or enzyme-linked immunosorbent assay. And in this case, we've plated down the alpha v beta 6 integrin into a 96-well plate and immobilized it. And then we compete that against serial concentration or serial dilutions of the diabody and cis diabody against a known concentration of fibronectin. And fibronectin is the natural ligand of alpha v beta 6. Once we've done this, then we can produce this nice binding curve, and what we're actually trying to calculate is the IC50, and this is the amount of diabody or cis diabody that is needed to displace 50% of the fibronectin. In this case, we got very, very good binding affinity with the cis diabody at 0.6 nanomolar, and almost identical with the non-covalent dimer, the normal alpha v beta 6 diabody at 0.8 nanomolar. So to give you an idea, anything in the sub-nanomolar range is ideal for pursuing in vitro cell binding assays, as well as moving on to preclinical and vivo studies. So we were pretty happy with these really low binding affinity results. Now to To verify the specificity of the two fragments, we run flow cytometry on them. And we have, in our lab, we have two engineered cell lines. One is the DX3 puro beta 6 line, which is alpha V beta 6 positive. And we have a DX3 puro line, which is alpha V beta 6 negative. The only difference between these two cell lines is the expression of the alpha V beta 6 integrin. So we then incubate both of these cell populations separately with both the diabody and the cis diabody, basically using those antibody fragments as primary antibodies to detect expression of alpha V beta 6. We then follow up with the secondary antibody, which is an anti-6-cis conjugated with lexaflor 48 to give us our fluorescent signal. We then look at if you look at the DX3 puro line here, is basically on the left column. You notice there's no shift in fluorescence with either the diabody, the cis diabody, or with our positive control. However, when you look at the DX3 puro beta 6 line, you notice a nice shift in fluorescence intensity with the diabody as well as the cis diabody, and these really nicely overlay with the 10D5 intact antibody, which is our positive control, and it's a standard that's known to be highly specific for the alpha V beta 6 integrin. So how can we convert these diabodies that we just produced into molecular imaging agents? And as I mentioned before, it's super important to keep in mind the biological half-life of your biomolecule and its relation to the radioactive half-life of your isotope. And in this case, the diabodies have roughly a biological half-life of around 2 to 4 hours in vivo. So this nicely pairs with F18, which has a half-life of 110 or 109.7 minutes. So in this case, we've synthesized the activated ester of 18F, which is fluorobenzoate, or SFB. And through mild conjugation reaction conditions, conjugated the activated ester to the exposed lysine residues on both the diabody and the cis diabody. We then purify these the crude reactions by spin columns and then inject them into a size exclusion column that's also connected to a PMT, or photomultiplier tube. And we use the PMT to detect the radioactivity that's present in the purified reaction. And you can see here on the right, this is a radio HPLC trace of the purified fraction, indicating a nice single radioactive peak belonging only to the purified radiolabeled diabody. We also want to determine whether or not the labeling using SFB affects the binding affinity and specificity of the particular diabody towards its target. So again, we're using the same engineered cell lines as we described earlier, the DX3 puro beta 6 and the DX3 puro lines. And we've then incubated the radiolabeled diabodies with these. We then, after the incubation time, we pellet down the cells and measure the radioactivity present in the cell pellet as that compared to the supernatant. And you can see here on the left with the diabody and the DX3 puro beta 6 line, we still retain almost 60% of the immunoreactivity even after radiolabeling it, with roughly 40 to 45% being internalized within the cell. As compared to the DX3 puro line, you get less than 2% binding. The cis diabody actually fared much better and still retained nearly 80% of its immunoreactivity towards the DX3 puro beta 6 cell line, with almost 70% being internalized. Again, with the same, with the DX3 puro alpha V beta 6 negative line, less than 2% binding. So this gave us a really good indication of not only was the binding affinity still retained, but also the specificity towards its target. So we were pretty happy with these results as well. So in conclusion, we've developed and produced two novel diabodies, or antibody fragments, an anti-alpha beta 6 diabody as well as a cis diabody, and demonstrated their potent and really selective binding to the alpha beta 6 integrin in vitro. We're still yet to do some in vivo studies, but those are upcoming, so we're planning on doing an in vivo small animal PET CT study. We have a mouse model that we use in our lab, again with the DX3 puro beta 6 and puro cell lines to generate tumor xenografts. And then when we radio label and inject the diabodies independently. So this will give us a good idea of how well does the in vitro results really translate to an in vivo study, and how well do these diabodies actually target the tumors in vivo. It'll also give us a good indication of how, with the blood clearance and pharmacokinetics of these particular compounds. In addition to that, we're going to conduct a biodistribution analysis on a separate population of mice. In this case, the mice are injected with the radiolabeled diabodies, and then instead of imaging them, we actually dissect them and remove the organs and then count the radioactivity in each of the organs. So this gives us an indication of the distribution of the radiolabeled compounds, and allows us to calculate the percentage of the injected dose per gram of tissue in each of the different organs, as well as the tumors. Finally, which is another interesting aspect of this project, is we're going to be performing site-specific labeling of the cis diabodies. As I mentioned, with the cis diabody, since it has the engineered disulfide bridge in it, it can be easily reduced. And what this allows you to do is attach the radiolabel or particular payload to the antibody fragment in a site-specific manner. And for antibodies, this is actually really important. Although we showed some very good affinity and specificity results after SFB labeling, it's very important that when you attach the radio label that it's not going to be interfering with the regions of the antibody fragments that are critical to antigen recognition and binding. So to do this, we actually engineered the C-terminus cysteine away from the binding regions of the antibody fragment and to allow site-specific labeling with copper-64 as well as with F18. So these studies we've actually done some copper-64 labelings. F18 is still in the works, but these are some of the future plans that we plan on conducting. With that, I'd like to acknowledge the members of the Sutcliffe Lab Group, as well as the staff at CMGI. The funding that comes from the Department of Energy, as well as my co-PIs at UCLA, Dr. Anna Wu and our postdoc, Kirsten Zetlitz. And at this time, I'd be happy to take any of your questions.
Thanks, organizer. My name is Yuan Pei. I'm a research faculty in the Department of Biochemistry and Molecular Medicine. My mentor is Dr. Keith Lang. And it's my honor to be here and share with you our research work on the smart and super versatile nanopolymorph platform. Our goal is to image and treat cancer using nanoparticles. So the concept of personalized cancer nanomedicine to leverage nanotechnology for cancer and patient-specific diagnosis and treatment. So developing multifunctional nanoparticles is very important for this goal. Based on our previous work on the telodymer-based drug delivery system, we recently synthesized this novel telodymer consisting of polyethylene glycol and a dendritic oligomer of cholic acid and porphyrin. So this polymer can self-assemble in aqueous solution into nanoparticles and load different drugs and create different metal ions in the core. We have demonstrated this nanoparticle system in the following applications. We can use this nanoparticle system for photodynamic therapy, photothermal therapy, near-infrared imaging. Of course, we can load the drugs for chemotherapy and load gadolinium for MRI and chelate copper-64 for PET imaging. I will show you in the following slide, one by one, of those applications. First, show you some of the characterization data of the nanopolymorph. After chelation with the metal ions, we can see significant shift in the absorbance spectrum of nanopolymorph. This is a TM imaging showing that the particle size of the nanopolymorph is around 20 nanometers in diameter. After chelation of the metal ion, in this case it's copper, we can see a higher TM contrast. Interestingly, the layer of the metal is in the interface of the core and the shell of the micelle of the nanoparticle, which we call the micelle. Because when the nanoparticle is stable in PBS, the near-infrared fluorescent actually is quenched. When we open up the nanoparticle by SDS, which is a strong detergent, we can restore the near-infrared fluorescence. Similarly, the ROS production under light irradiation from this nanopolymorph is also minimum in PBS, but we can restore it in the presence of SDS when we open up the nanopolymorph and the polymorph becomes dequenched. Interestingly, we found that when the nanopolymorph is quenched in PBS, usually the energy after the light irradiation released in the form of heat. So this is a drop of nanopolymorph solution. We can use laser to heat it up to 60 degrees, which we can use for the photothermal therapy. We have also demonstrated we can load a whole bunch of drugs in this nano-porphyrin platform. For chemotherapy, the particle size of the drug-loaded nanoporphyrin is remains the same particle size around 20 nanometer, which is optimal for tumor targeting. We can also use our cross-linking strategy to make an even stable nanoparticle. Due to the time limit, I won't go to the details. The first application I want to show is to use the nanoporphyrin for near-infrared fluorescence imaging. This is a transgenic mouse with spontaneous breast cancer. There are 9 tumors. You can see all the tumors could be lit up by nanoporphyrin, while the normal organ remain very low signal. Interestingly, we took the lung of the animal out. This is a small piece of lung tissue, but there's a tiny metastatic lesion in the lung. We can see the nanoporphyrin can also go to the metastatic side of the near the lung. So this is very important because we can use this formulation to detect the small tiny metastatic lesion, and also can deliver drugs to treat metastatic disease, which is a very hard topic in cancer research. We have developed a different tumor targeting peptide ligand in the lab. So in this case, we decorate nanoporphyrin with PLD4 ligand, which is a bladder cancer targeting ligand. And we incubated the targeting version of nanoporphyrin with bladder cancer cells together with the normal urothelial cells. The bladder cancer cells has the green fluorescence here. We can see the nanoporphyrin can selectively go to the bladder cancer cells, not the normal urothelial cells, demonstrating the tumor selectivity of this formulation. We then established our blood cancer model. This is our orthotopic blood cancer model, BL269, and we inject nanoporphyrin with ligand intravesically. You can see the nanoporphyrin can light up blood cancer, but not normal bladder. This is a section of the bladder. We can also see the nanoporphyrin can go to the cancer side, but not the normal bladder. Interestingly, we found a very interesting result. So here, we did the injection, and then we performed the imaging without wash away the nanoporphyrin, and the unwashed nanoporphyrin left a very low background because it's quenched when it's stable. But once it go to the tumor site and become dissociated, it gives you a fluorescence at the tumor site, which will prove our idea to use this nanoporphyrin formulation to do low background image, low background near-infrared image. Another application of this formulation is to do MRI as an MRI contrast agent. So here we load gadolinium in the core of the nanoporphyrin. This is a TM picture showing that the morphology. The idea of using the nanoporphyrin gadolinium formulation for MRI is when the gadolinium is the core of the nanoparticle, it has a little chance to affect the proton nearby. That's why during the blood circulation, when the nanoporphyrin is stable, there's a very low MRI signal enhancement. Once it goes to the tumor site, especially inside go to the tumor cells, it becomes dissociated. The gadolinium has increased ability to affect the surrounding proton. That's why it gives you a high signal at the tumor site. So we performed our in vitro study showing that when the nanoporphyrin GD formulation is stable in PBS, it gives you very low MRI signal enhancement. But once you use SDS to open the nano-formulation, you can see significant higher MRI enhancement. We also performed our in vivo MRI, showing that the nano-polymer GD formulation can light up the tumor, and the signal in the rest of the body remains low. We also performed our SCP-MS to quantitatively look at the GD biodistribution. We found a significant tumor uptake of the gadolinium, showing that the excellent tumor targeting of this formulation. Another application of this nanopolymer is to do packaging. We can efficiently kill copper CD4 in this formulation in 40 minutes. And when we inject the copper nanopolymer in the mice with SCO-3 ovarian cancer, you can see at 16 hours a significant tumor accumulation of this formulation. And the combination can retain the tumor site up to 2 days, while the rest of the body, has a very low signal, give you very good contrast between the tumor and the normal tissue. So I just showed you a few of the imaging application of this formulation, but how about therapy? So this is a transgenic mouse with breast cancer. This is a tumor here. So once we apply the nanopolymer by IV, this is actually the 1 day later we use the laser to shine on the tumor site. We can see significant heat generation at the tumor site, for photothermal therapy. We also take out the tumor tissue. We found a significant singular oxygen production at the tumor site. Basically there are two modalities: the heat generation and also the ROS generation. So we call it dual model phototherapy. Basically, we can combine the two therapeutic modalities in one formulation and treat the tumor. We then performed our in vivo study to look at the anti-tumor efficacy of this formulation. After 3 doses, you can see the tumor inhibition is very significant. After 3 doses, actually we can almost eliminate all the tumors in the transgenic mice with spontaneous breast cancer, which is very impressive. Since we can use this formulation for imaging and also therapy, and why don't we combine the imaging modality with the therapeutic modality and do our imaging guided therapy? To prove the idea, so we use the gadolinium chelated nanopolymer, inject it into the mice with tumor, and use the MRI to follow up the tumor growth. We can see at 4 hours, we can see the significant tumor signal of the nanopolymer gadolinium, and 1 day later, we shine light on the tumor site to perform the therapy, the phototherapy, and then we continuously use the MRI to monitor the tumor growth. And after 24 hours after the light, we can see large area of the necrosis inside the tumor, as indicated by the MRI image. And we found that at 7 days later after the therapy, the tumor was totally eliminated. So this is really showing the formulation can combine the different imaging and the therapeutic modality in one formulation. So to summary, the nanopolymers can be used as a nanoprobe for multimodal imaging, including near-infrared imaging MRI and PET, nanocarrier for drug delivery. I didn't show you the function here, but I have some in vivo data showing that it's also very efficacious. The nanopowder can also be used as a nanotransducer to convert light to heat and rocks for dual mode phototherapy. And we'd like to make an acknowledgement. This work is done in working with Dr. K. Lam in biochemistry and molecular medicine, Dr. Han Chongxian in nuclear medicine, and our collaborator are from CBST, Dr. Frank Changhin, and also from BME here, Dr. Sam Cherry, Dr. Laura McHugh, and Kathy Ferrara. Of course, Jeff Walton. Dr. Jeff Walton helped us a lot with the MRI. And finally, the funding agency. And I would like to stop here, and I will be happy to take questions.